Alright, so that's the stomach, and now our chyme is in our small intestines. So let's talk about the function of the small intestine. Just really quick, anatomically, remember your small intestines extends from your pyloric sphincter to your ileocecal valve, and you have the three regions. You have the first part is your duodenum, and overall the function of your small intestines is to complete your chemical digestion, get all your macromolecules into their monomers down to the simple sugars, amino acids, and fatty acids so you can absorb them. And most of your absorption of nutrients happens in the duodenum. So the first part was your duodenum. That's where your chemical digestion is completed, and then the absorption. The middle portion of the small intestine is called the jejunum. You have some absorption that's happening there, but not as much. And then the last part is the ileum, which joins with your large intestines. In the ileum, you do absorb your vitamin B12, utilizing that intrinsic factor, and some cholesterol will be absorbed, and we'll talk about that. But overall, most of your absorption happens in the duodenum, maybe a little if it's left over in the jejunum, very little absorption or digestion in the ileum. If we look again at the histology of your small intestines, it's basically designed to maximize the absorption. If you want to maximize absorption, you need to increase your surface area. So the way we do that is, remember, you have the folds on the lumen, which are your plica circularis. Hopefully you remember that from anatomy. And then you have little finger-like projections along the lumen, which are called villi. And then if you look at one single villus, which you're looking at over here, remember each villi is lined by simple columnar epithelium. So you're seeing the simple columnar here. And if you were to look at one of these simple columnar cells closer up, like you see here, on the apical surface of that epithelial cell, there's little folds, and those little folds are called microvilli. You're increasing the surface area quite a bit by the plica circularis, the villi, and the microvilli on the epithelial cells. For absorption to occur, in the connective tissue under the epithelium, in the submucosal area, you have blood vessels, and you also have a lymph vessel, which is called in the small intestines a lacteal. So what will happen is the nutrients, for it to be absorbed, needs to pass from the apical surface of the epithelium through the epithelial cell and then into the blood or the lymph. Okay, so the nutrients is going to be absorbed through this epithelial cell. Now when the chyme enters into the duodenum here, now when the duodenum curves around the head of the pancreas, and then remember also you have your bile ducts that connect to the duodenum. And so when chyme enters into the duodenum, bile from the liver will be emptied into the duodenum there, and digestive enzymes will come from the pancreas, which empty into the duodenum to aid with digestion. So we're going to talk about the function of bile in the liver in digestion, and then we'll talk about the digestive enzymes that come from the pancreas, how they play a role in the duodenum to complete digestion and absorption. So let's start with the liver and bile production. So the liver has many functions. They actually say that the liver has over 200 different functions. We're not going to learn about the 200 different functions. You just need to know a few of them. Some you already know. We learned that the liver stores excess nutrients and then releases them into the blood when you need them. For example, when you have extra glucose, remember the liver stores it as glycogen, and then when blood levels drop, it will dump the glucose into the blood. It does the same thing with some vitamins and nutrients. The other thing the liver does is it will detoxify any drugs or metabolites. That's why you have the hepatic portal system. So if you absorb any drugs, metabolites could be byproducts of metabolism. They go to the liver and the liver cleans them out. We already learned that the liver produces a lot of your plasma proteins. So that's why people that like all your clotting proteins, angiotensinogen, different proteins, that albumin that are in the plasma are produced by the liver. That's why we learned that people that go into liver failure can have clotting problems. And then today we're going to add on to our list of functions of the liver that we know about by talking about how the liver produces bile. And bile's role is to aid in the digestion of fats. So you should remember this from anatomy. Remember your hepatocytes, and remember your liver lobules, and you have the hepatic portal vein that comes in with the artery and leads into the sinusoids that then empty into the central vein. and as the blood goes past the hepatocytes, they adjust it and put proteins and different things into it. While on the other side, they're doing another function. There's bile canaliculi, or little bile canals. So these hepatocytes are producing bile and putting it into these little canals, and all the canals drain into bile ducts. And eventually, all those little microscopic bile ducts drain either into the right or left hepatic duct from the liver. And that bile will travel down the common hepatic duct into the bile duct, common bile duct, and it goes into the duodenum. Well, here at the hepatopancreatic ampulla, there's a sphincter where it meets with the pancreatic duct from the pancreas. Well, there's this other sphincter here called the sphincter of Oddi, which is at the base of the common bile duct, and it prevents bile from getting into the duodenum when there's not any chyme there, because you don't need the bile to go in there when the duodenum's empty. So that sphincter is closed, but the liver keeps making bile, so bile starts to back up the duct, and it gets stored in the gallbladder. And the gallbladder will also start to make it more concentrated. The longer it sits there, the more concentrated it gets, because the cells will absorb water. And that can be kind of a problem, because the longer it sits there, the more concentrated it can get, and bile has cholesterol in it, and that cholesterol can crystallize and cause stones, and that's where you get the gallstones. Okay, and those gallstones can be painful because they can get trapped in the ducts, or when the gallbladder contracts, you feel those stones. There's certain people that tend to be at a higher risk for gallstones. It's mostly middle-aged women, like in their 30s, 40s, or younger, that tend to get gallstones. And the reason is estrogen plays a role in the amount of cholesterol that gets put into bile. So women tend to have more cholesterol in the bile than men, because it has to do with the estrogen. The other thing they've seen is that the gallbladder isn't emptied at a regular rate, like people that don't eat regular meals, or they've seen a higher risk of people that diet a lot, having a higher incidence of gallstones, or it can completely be just genetic or just luck. And they can remove your gallbladder, and you're still okay. You can still live, and most of you are aware of that. And the reason is they keep this duct here from the liver, so you can still produce bile, but you can't store a large amount of it. So sometimes you have problems with digesting high fatty meals, so you kind of have to adjust your diet. So let's look at the function of bile. Bile aids in mechanical digestion of fats. What will happen is, you guys know that lipids do not mix with water. They're hydrophobic. And so you get these large fat globules, or you've seen when you've mixed oil with water, all the fat comes together and you get one big fat globule. Now, if you were to start to try to digest that big globule with enzymes, you have very little surface area to work on, surface area to volume. So it would take you a long time to break down that fat globule with the enzyme lipase. So what bile does is it emulsifies the fats. It breaks that fat globule into smaller little globules, so that's called emulsification. And you can see here, if you have smaller globules, now they have a larger surface area to volume ratio that the enzyme lipase can start to work on, so it helps speed up the digestion of fats. Remember we talked about amphiphilic molecules, where they have a polar end and a non-polar end? That's how bile works. It has a lipid-soluble portion that's made out of cholesterol, or cholesterol-based, and then a water-soluble portion that's charged, or polar. Okay, so it has a polar end and non-polar end. So the non-polar end will orientate itself towards the lipid, and the polar end will orientate itself towards the water, and it breaks the molecules into smaller globs. So what you'll find in bile is basically aqueous. It's a watery solution. It's slightly basic to help neutralize the chyme coming from the stomach. And then you have the bile salts, which we talked about has a non-polar cholesterol-based steroid region and a polar region. You'll also find cholesterol in the bile. And there's another molecule called lecithin, which is a phospholipid. And remember, a phospholipid is amphiphilic too, so it helps. It has the polar heads and the lipid tails. And the other thing is bilirubin. Remember, we learned about this in our blood chapter, that when red blood cells are broken down by the spleen, a byproduct is bilirubin. And that bilirubin needs to be removed from the blood, otherwise it will deposit in like the skin or the tissue of the eyes, and you get what we call jaundice. The bile is one way you get rid of bilirubin. The other way is your kidneys will put it into the urine. So what controls the release of bile? Well, it's basically the stimulus is when chyme gets put into the duodenum. So when the duodenum detects acidic fatty chyme, it releases the hormones, which you already learned about, cholecystokinin or CCK and secretin. And what secretin does is it works on the liver, and it triggers the liver to produce more bile. And cholecystokinin, this is what chole refers to, and cysto refers to gallbladder. And so it was actually named for this function, even though it has other functions like on the stomach. But cholecystokinin causes the gallbladder to contract and the sphincter to relax so that the bile gets dumped into the duodenum. So the stimulus for bile production and release is the chyme entering into the duodenum, and then you release these hormones. The other thing is the vagus nerve, which is parasympathetic, can also cause contractions of the gallbladder. So that's what this picture is showing. Basically, chyme gets dumped into the stomach, chyme from the stomach gets dumped into the duodenum, and those enterocells of the duodenum will release their hormones, cholecystokinin and secretin. Secretin will work on the liver and cause it to produce more bile. And then the cholecystokinin will cause the gallbladder to contract and release the bile into the duodenum. Also, the vagus nerve, which is parasympathetic, can cause contractions of the gallbladder. So bile will travel through the small intestines. Your body likes to recycle. So a lot of the bile salts will end up getting reabsorbed in the ileum and get put back into the blood and travel back to the liver and be recycled. About 5% of the bile will escape in feces, so it will be lost. So there is some drugs for cholesterol, to reduce cholesterol, that inhibits the process of reabsorption of the bile salts in the ileum. Because the idea there is kind of you can dump more cholesterol through your feces if the bile stays in the intestine and doesn't get reabsorbed. There's other ways of reducing cholesterol. That's just one class of drugs that works that way. So now the other organ that's producing a lot of the secretions that get dumped into the duodenum is the pancreas. And there's two things the pancreas does. It makes a lot of the digestive enzymes, which we'll learn all of those. And the other thing it needs to do is its secretion neutralizes the acidic chyme. The acinine cells produce the digestive enzymes, and then there's another cell in the pancreas called duct cells that secretes sodium bicarbonate. Okay, and the bicarbonate, it's basic. It's alkaline. And so it will neutralize the chyme when it gets in the duodenum. Because the duodenum isn't designed like the stomach. It has some mucus, but not as much. And so the chyme needs to be neutralized. So what regulates pancreatic secretions? Guess what? It's the same two guys. CCK, cholecystokinin, and secretin. So when the chyme gets put in the duodenum, these hormones get released, and they travel to the pancreas. So CCK triggers the acinine cells to produce the enzymes, and the secretin works on the duct cells to increase the secretion of the bicarbonate, or the sodium bicarbonate. And then again, you have your parasympathetic nervous system, which causes the release of the pancreatic juices. So, you've learned already that CCK and secretin, they work on the pancreas, okay, to increase their secretions. They work on the liver and gallbladder to increase the production of bile. And then they work on the stomach to actually inhibit the stomach secretions. Okay, so they activate the gallbladder and liver, they activate the pancreas, they inhibit the stomach secretions. And then you've also learned that the vagus nerve will stimulate secretions of all digestive secretions. So stomach secretions, pancreatic secretions, salivary secretions, gallbladder and liver, bile production. Parasympathetic would increase all those secretions. And this is just a picture showing you that the cholecystokinin and secretin get dumped in the blood, and they work on the pancreas. Here's another summary of the intestinal phase. So what happens when the chyme enters into the duodenum? Okay, so here's your stomach phase, small intestines, and then what's happening in the pancreas. So food enters in the stomach. When food enters in there, you increase the secretions, acid secretions, enzyme secretions, gastric motility increases. When the chyme gets put into the small intestines, it's going to inhibit the stomach secretions and motility. So we just talked about all the fat, acidic, hyperosmolarity, and the hormones that it releases, CCK, secretin, will go back and inhibit gastric motility and secretions. And then how does that relate to the pancreas? Well, when the chyme gets put into the small intestines, remember your gastroinhibitory protein, I told you that will trigger the pancreas to release insulin? Because the idea is, okay, we're going to start absorbing glucose, so we need to get insulin into the blood. And then we just learned that CCK will increase enzyme secretions of the pancreas, and secretin will increase the bicarbonate secretions of the pancreas. So this little chart is just summarizing what's happening in the stomach, intestines, and how that relates to the pancreas. Alright, so let's look at the role of all the pancreas enzymes that are released. So all the enzymes that get released by the pancreas is to help further along chemical digestion of your proteins, carbohydrates, and lipids. So first let's talk about the enzymes that play a role in proteins. Proteolytic enzymes. Proteolytic is a general term for enzymes that play a role in protein digestion. And there's a lot of them. All these different proteolytic enzymes each break at a different peptide bond. I remember I had to learn in biochemistry which bonds each of these break. To be honest with you, I've forgotten. Okay? I'd have to look it back up. And you're not going to need to learn that either. But the idea is there's 3 different enzymes, or there's more than this, but these are the main ones. Trypsin, chymotrypsin, and carboxypeptidase. These 3 enzymes break peptide bonds between amino acids. For example, I'm making this up right now, but trypsin, let's say it cuts between wherever along on the protein, wherever you see a bond between leucine and valine, those 2 amino acids, it would cut there. And then chymotrypsin recognizes different bonds. Maybe he recognizes a bond between glycine and aspartic acid, and he'd cut there. The idea is each of these enzymes cut at different peptide bonds where it sees certain amino acids, and so they chop it up like scissors, and eventually you get shorter peptide fragments. The pancreatic enzymes, the proteolytic enzymes, are all going to be secreted in their inactive form, and it's the same reason. You don't want to dump these proteolytic enzymes into the pancreatic ducts and have them start degrading the pancreas. So they don't get activated until they get into the duodenum. So in their inactive form, the pancreas secretes trypsinogen, see the GEN, chymotrypsinogen, GEN, inactive, and then newer named enzymes, instead of doing the GEN, the enzymes have the ASE on the end. So if it's inactive, they put a pro in the front. So procarboxypeptidase is the inactive form. It's just newer terminology. And they'll get activated in their active form. Trypsin with an IN, chymotrypsin IN, and then you just drop the pro, and it's carboxypeptidase. And then how they get activated is by other enzymes. Okay, so chymotrypsinogen, once it gets dumped into the small intestines, it gets activated by the enzyme enterokinase into trypsin. Let's look at this picture. So you dump the inactive forms in, trypsinogen, chymotrypsinogen, procarboxypeptidase. They're in the small intestines. Now there's an enzyme that's in the membrane of the small intestines called enterokinase. So it's a membrane-bound enzyme, or we call those brush border enzymes. And this enzyme activates the trypsinogen into trypsin. And then trypsin is the enzyme that then activates the other 2 into their active forms. So those are proteolytic enzymes, 3 of them, that start breaking proteins down into smaller peptide fragments. Okay, so you're continuing your protein digestion. You also have to continue your carbohydrate digestion, so you have amylase again. So you have salivary amylase and you have pancreatic amylase. They do the same thing. It's just who's secreting them. So amylase, remember, breaks your polysaccharides down into disaccharides. You also, if you're eating any animal or plant product, which hopefully most of our food is, if you're taking in cells, either plant or animal, they're going to have DNA and RNA, or nucleic acids. So you have an enzyme called nuclease, which hydrolyzes or breaks down the nucleic acids into their individual nucleotides, A's, G's, T's, and C's. And then you have lipase, which is going to chemically change triglycerides into either monoglycerides and fatty acids. So you have your triglyceride, which had a glycerol and 3 fatty acids, and it's going to break it down into free fatty acids and a glycerol. There's other types of fats you can take in besides triglycerides, but that it's basically going to break them all down into fatty acids. So we're down to disaccharides and small peptide fragments, but you still have to get down to the monomers. And so the final chemical digestion is carried out by enzymes we call brush border enzymes. And they're called brush border because if you look at the epithelial cells lining the lumen of the small intestines, remember they have microvilli on them. So the microvilli they call the brush border because it looks like a brush, like a hairbrush. Or fringe, I think of it as fringe. And so there's enzymes that are actually embedded in there. They're not being secreted by the pancreas. There are enzymes that are already in the small intestines that are embedded in that membrane. There's a number of them that break the disaccharides into monosaccharides. So if you have maltose, which is a disaccharide, there's the enzyme maltase that breaks it down into glucose and glucose. Or if you have sucrose, sucrase will break it down into its monosaccharides. And if you have lactose, which is glucose and galactose stuck together, the enzyme lactase will break it down into those simple sugars. So when you hear people are lactose intolerant, that means that they have trouble breaking down the disaccharide sugar lactose because they don't produce very much of this lactase. And as we all get older, we produce less lactase. But some people, just genetically or by chance, tend to produce less of this than most of us. So you'd say they're lactose intolerant. And what happens is that lactose doesn't get broken down, and it will get into the large intestines. And you have a lot more bacteria living in the large intestines than you do in your small intestines. And when that sugar gets to the large intestines, the bacteria love it, okay? And they'll eat it and utilize it. Well, bacteria don't really eat, but they're going to utilize it for metabolism, and a byproduct of their metabolism will be gas. And that's why it causes people that are lactose intolerant, if that sugar stays in there, they start to have more digestive problems. And then there's another enzyme that's going to finally break down all your peptides, your small peptide fragments, into the individual amino acids. And that brush border enzyme is called aminopeptidase. So here's a summary. You have your polysaccharides like starch or glycogen, and amylase starts to break it down into its disaccharides, such as maltose. And then you have the brush border enzyme maltase, which finally breaks it down into glucose and glucose, its monomers. Okay, and then they're showing it for sucrose. It would be broken down into glucose and fructose, and lactose would be broken down into glucose and galactose. So amylase is produced by the salivary glands in the pancreas. These three are brush border enzymes that are found on the border of the small intestines. And then you can see here with proteins, you start protein digestion with pepsin in the stomach, and then you have your other proteolytic enzymes that get secreted by the pancreas, the trypsin, chymotrypsin, carboxypeptidase, and they're just chopping it at certain amino acid sequences so that you get smaller peptide fragments. And then the brush border enzyme aminopeptidase will finally break it down into the individual amino acids for absorption. So let's say we got everything digested into the monomers and we're ready to absorb the nutrients from the small intestines into the blood or lymph. So most electrolytes, nutrients, and vitamins are completely absorbed. I already mentioned that your digestive tract is pretty efficient at absorbing everything. We're going to briefly touch on these. Vitamins, if they're fat soluble, okay, for example, A, D, E, and K, they're absorbed along with lipids. So they get dissolved into lipids, and when you absorb the lipids, you get your vitamins. Water-soluble enzymes like C and B are absorbed with water. And we talked a little bit briefly about how vitamin B12 has to combine with that intrinsic factor that is produced by the parietal cells in the stomach and get absorbed by receptor-mediated endocytosis in the ileum. If we look at a few minerals, just to give you examples, we're going to look at calcium and iron. There's other minerals, but we'll focus on these two because they're kind of your most important. They are absorbed on an as-needed basis by active transport. So what that means is you won't just absorb all the calcium you take in, you just absorb it on how much you need. for the most part. So calcium absorption will depend on blood levels of ionic calcium. And that's going to be regulated by the parathyroid hormone, which you already learned. And we talked about how you require vitamin D for the absorption of it. And that's why they put vitamin D in milk. People that live in sunny areas, you get vitamin D from the sun. Well, you produce vitamin D in response to the sun. But people that were living in areas where it's darker weren't producing enough vitamin D, and they were becoming calcium deficient. Now, iron also is absorbed on an as-needed basis. But what happens is it gets absorbed and it gets stored in the epithelial cells of the small intestines. And then as it's needed, it will get transported into the blood and carried on. We learned this when we learned blood, the protein transferrin, and it will go and help with the production of hemoglobin. Some of it gets lost when you do shed the lining of this epithelium, so it can be lost when the cells are slothed off, but for the most part it's getting stored there and then transported in the blood. Now, electrolyte absorption, for example, chloride, and then some of your nutrient absorption, such as glucose and amino acids, they all depend on the active transport of sodium into the interstitial fluid. You learned this concept back when we learned about secondary active transport. So basically, through active transport, you're creating a sodium gradient, and chloride will follow the sodium because it's a negative ion. So it's an anion that will follow the positive ion. And water will always follow the sodium because we know that water follows salt down its osmotic gradient. So the idea here is in the cells of the lumen, you have active transport of sodium that's creating the gradient. Now when you look at sugars and amino acids, that's through secondary active transport. If I create a sodium gradient, where sodium is always low in the cell because I'm pumping the sodium out using primary active transport, then I have a co-transporter that transports sodium along with glucose, or it could be sodium along with an amino acid. Okay, and as sodium moves down its concentration gradient, it brings glucose with it, despite the concentration of glucose in the cell. And then the glucose and the amino acid will enter into the capillary, and it'll be transported to the liver through the hepatic portal vein, where, if you have too much glucose, it will be stored as glycogen, or it will continue in the blood. So this is kind of a better picture for secondary active transport. This is the lumen of your small intestines, where the chyme is. Okay. And these are the cells that are the epithelial cells lining the lumen, and then in the submucosal area, you'll have your capillary. So you need to get your sugars from the lumen through the epithelial cell and then into the blood. So you have your sugars, they'll get broken down by amylase into the disaccharides, and then you have brush border enzymes that are sitting in the membranes of those epithelial cells that will break them into their simple sugars. And you have a sodium glucose transporter, sodium glucose transporter that's going to bring the glucose in with the sodium. So you have to create a sodium gradient. So down here at the basement membrane, you'll have a sodium potassium pump that requires energy because that's primary transport. It requires ATP. So you're constantly pumping sodium out of the cell. So what that does is it creates a gradient that drives sodium into the cell on this side. And as it drives sodium in, glucose hitches a ride on the co-transporter. So this is your secondary active transport. So what this means is even if glucose is high in the cell, you'll keep driving glucose in. So you're really efficient at absorbing glucose. You absorb almost all the glucose that you take in, which is scary. Same thing with amino acids. You have a primary transport here that requires energy, creating a sodium gradient because you're pumping sodium out of the cell. So sodium is driven into the cell on this side, and it brings amino acids with it. And then they'll just diffuse by diffusion from their higher concentration in the epithelial cell into the blood. Now lipids are lipid soluble, so they don't require a transporter on the cell surface. But what happens is fatty acids and monoglycerides, they form little globules called micelles or micelles in the lumen. And they're lipid soluble, so they will just fuse with the plasma membrane, and they will travel to the endoplasmic reticulum. And the endoplasmic reticulum, or specifically the smooth endoplasmic reticulum, SER, will take those fatty acids and glycerol and make them back into triglycerides. So it's kind of odd. Lipase, that enzyme in the lumen, will break the triglycerides down into fatty acids. And then when it gets into the cell makes it back in, takes fatty acids and makes them into triglycerides. They'll go to the Golgi, where a lot of times they'll be associated with proteins, because lipids can't travel in the blood unless they're necessarily associated with a protein, because they're not water soluble. So they're combined with proteins to form what we call chylomicrons, and then they're released by exocytosis into the submucosal area. And most of your fats, some of your fats will be transported in the blood, but a lot of them get into the lymph system, so they get into the lacteal that's in the villi. And eventually they'll get dumped into the blood when the lymph system empties into the subclavian vein. So we just talked about all the enzymes and how you finish chemical digestion in the small intestines, and then how you absorb the nutrients in the small intestines. Now we're going to talk a little bit about the motility, how you move things along the small intestines. So the small intestines carry out segmentation. Okay, it's the most common motion of the small intestines. It gets initiated by pacemaker cells in the smooth muscle, and it spreads through gap junctions and causes the smooth muscle to contract together. And the purpose of the segmentation in the small intestines is to mix the chyme with all those digestive juices that are coming from the pancreas, and also the bile that's coming from the gallbladder. And the other thing it does is, as you do segmentation, it's exposing the chyme to the surface area of the small intestines to enhance absorption. In the small intestines, segmentation is occurring at a faster rate at the duodenum than it is at the ileum. So at the proximal end, it's occurring a little faster than it is at the distal end. And so what that creates is it's steadily moving things towards the ileum. Things that will enhance the activity of the smooth muscle in the small intestines, therefore enhance the intensity of segmentation, is you have distension, so you have stretch receptors, gastrin, which got released by the stomach, and extrinsic nerves such as your parasympathetic nervous system. So all these things are going to increase the activity of the smooth muscle. Now once all the nutrients has mostly been absorbed, and the way that it can tell that most of the nutrients has been absorbed is it's detecting the hypotonicity of the chyme. So a lot of times once all the solutes have been absorbed, then the small intestines knows, okay, we're pretty much done with absorption. I can move things along and send it to the large intestines. So what happens is, the way it moves it is it will trigger, after segmentation and mixing and absorption, it will trigger a series of peristaltic waves called migrating motility complexes that begin distal to the previous. So it starts at the duodenum, it moves food forward, and then it starts a little further along, moves stuff forward, and so forth. And it's basically, its purpose of the migrating motility complex is to move the food, or whatever's left from your meal. Okay, there's some bacteria that are in there, mucosal cells that you've shed, move it all into the large intestines. And what triggers it is during your post-absorptive state, so after you've absorbed everything, the endocrine cells that are in the mucosa of your small intestines will release a hormone called motilin. And that motilin is a hormone that triggers the migrating motility complex. And like I said, the endocrine cells, what stimulates them to release the hormone is they're detecting changes in solute concentration. So once the food gets to the ileum, or what's left over, it needs to get put into the large intestines. And it's going to be emptied into the cecum of the large intestines through that ileocecal sphincter. I call it the ileocecal valve, and it's a one-way valve. So things can only go from the small intestines into the large. They can't come back. And that's important because there's a lot of bacteria living here in the large intestines, that if it was able to get into the small intestines where you still have a lot of nutrients, it would just flourish and cause you problems. So there's a reflex called the gastroileal reflex. So gastro refers to stomach, ileo refers to the ileum. And so what happens is when a new meal comes into the stomach, it triggers, remember, the release of gastrin. And that gastrin hormone will inhibit the ileosphincter and allow the food, that valve to relax and food move into the large intestines. So it's a feed-forward mechanism. If I have a new meal coming into my stomach, I need to make way for it. So I need to move everything from the small intestines into the large intestines. So now we're into the large intestines. And we talked anatomy-wise, it frames the small intestines. You have your ascending, transverse, descending, sigmoid colon. And so it extends from the ileocecal valve to the anus. And its main functions in the large intestines is you've already absorbed most of your nutrients, but what you will absorb is any remaining water. So the fluid chyme is now going to become more solid. You need to reabsorb the water. You will reabsorb some of the remaining electrolytes like sodium, potassium, and so forth that are still dissolved in the water. And then you do absorb some vitamin K that is actually made by the bacteria that live in your large intestines. And the other function is obviously to get rid of any indigestible food, so it eliminates it from the body. Now the motility in your large intestines, how it moves things along, is it's a lot less active than your large intestines. It will do segmentation, but it only occurs about 3 to 4 times a day. It usually correlates with your meals. Where I think, I'm trying to recall this, so don't hold it to me. I should have double-checked before I started this lecture, but I want to say that the small intestines, it carries out segmentation about 12 to 13 times per hour. Okay, so it's more active than the large intestines. And there's a reflex called the gastrocolic reflex. So what does gastro refer to? Stomach. Colic refers to large intestine. So it's a reflex between the stomach and the colon. And what happens is, again, when a new meal enters into the stomach, you release gastrin from the stomach and also some of your extrinsic nerves, which would be your parasympathetic nervous system. It will trigger this reflex that will cause the muscle of the large intestines to contract. And what happens is the ascending and the transverse contract, and there's like a mass movement of the content into the rectum. So it's kind of one big movement. It will do segmentation a few times a day, and then when a new meal enters, you have this mass contraction that forces the food, or what's left, the indigestible material, into the rectum. And that will trigger a reflex. So when the food moves into the rectum, there's stretch receptors in the wall of the rectum, and it triggers what we call the defecation reflex to eliminate the food from the body. The rectum stretches from the mass movement, and that gets detected by a sensory neuron through the just a sensory neuron that travels to the central nervous system, and then it's a parasympathetic reflex, autonomic reflex. The output comes back and it innervates the smooth muscle of the rectum, and it also controls, you have voluntary control over your external anal sphincter. So hopefully you remember this from anatomy. In anatomy, you should have learned you have an internal anal sphincter, which is smooth muscle, which is involuntary. You can't control that. And then you have your external anal sphincter, which is skeletal muscle, which you do voluntarily control. So the autonomic reflex, the parasympathetic nervous system, will make the smooth muscle of the rectum contract and relax the smooth muscle of the sphincter so that you can eliminate the undigestible waste. But you can voluntarily then relax the external anal sphincter so that you can eliminate it when it's convenient for you, basically. And that's what I'm going to end on, the defecation reflex. But that's your path through the digestive system. Okay? That's it for now.